Okay, today we're going to talk about the dihybrid cross. You guys have been working with monohybrid crosses. Mono, one. We're talking about one trait at a time. Now we're going to talk about two traits at a time. Because when Mendel ran his experiment, he did his P1, pure crossed with pure. He did his F1, where he crossed heterozygous with heterozygous. His F1 cross then gave him his F2. And in all of those, he was just looking at one trait. After he did that, he decided he wanted to look at two traits to see if they would affect each other. Would the height of the plant affect the color of the plant? Would the color of the seed affect the color of the plant? That type of thing. So then he ran experiments looking at two traits at a time. And those he called dihybrid crosses, which is a breeding experiment that tracks the inheritance of two traits. From his experiment, he came up with the law of independent assortment, which means that each pair of alleles segregates independently during gamete formation. So big T, little t, crossed with big T, little t, the big T and the little t are going to separate on their own. They won't affect each other, and they're not going to affect any other genes. So the genes for height do not have any influence on the genes for color, or your genes for hair color do not have any influence on your genes for eye color. And then there's a formula to figure out how many different gametes a person can produce. It's 2 to the n. Here's some examples. And you want to look at the number of heterozygotes in the genotype. So in number 1, we have 2 heterozygotes. So 2 to the second, 2 times 2. So that person would produce 4 different gametes. There's all the possibilities. And I'm going to show you how to figure out the possibilities later. If we were ever looking at something like this, here we'd be looking at 4 traits, the A trait, the B trait, the C trait, the D trait, or maybe height, seed color, pod color, seed shape. We will never do anything like that, but this is just an example. And if you look at this, in this genotype, you have 3 heterozygotes, so 2 to the third, or 2 times 2 times 2, this person would produce 8 different types of gametes. There's all the possibilities. And this is definitely something we wouldn't do, but if you look through here, there's 6 heterozygotes, 2 to the 6, you're going to have 64 gametes. Okay, let's say we're looking at seed shape and seed color. There's all the possible alleles. The R is for the shape, the Y is for the color. You know round is dominant over wrinkled and yellow is dominant over green. So let's do a cross. This would actually technically represent Mendel's F1 cross because you have heterozygous crossed with heterozygous. So again, Mendel's F1 cross is always a heterozygous crossed with a heterozygous. So for that first plant, there's all possible genotypes. For the second plant, there's all possible genotypes. Okay, so here's a cross. I'm going to show you how to actually do the Punnett squares, how to get all possible gametes. You need a box that has 16 squares. And the easiest way to do this is you draw a big box. Then you'll want to draw a line down the middle, and a line on top, and a line on the bottom. Then divide it down the middle the other way, and then divide each of those in half. And I've color-coded the top row. So we have our first plant, big R, big Y, little y, crossed with the big R, big Y, little y. So let's just say that this is the mom, and here's the dad, and we're going to put mom's genes here, and we're going to put dad's genes here. Okay. So remember, when you're making eggs and sperm, you get half of every gene. So across the top are going to be the eggs because that's going to represent the mom. Down the side are the sperm. So you can see we only have one R and one Y in every spot because it's representing the sex cells. So in this case, what you want to do, you want to mix all R's with all possible Y's to get all possible gametes, or all possible eggs. So what you would do, you would take this first R and mix it with the first Y. So you can see we have big R, big Y. Then you want to take that first R, but now you're going to mix it with the second Y. Big R, little y. So we've taken that first R and we've mixed it with all possible y's. Now take the second R, mix it with the first y, so you have little r, big Y. Then you take the second R and you mix it with the second Y, little r, little y. And you would do the same thing down the side. If you ever are doing a large Punnett square and you see across the top or down the side that you put two of the same letter, it is wrong. Now, once you got them across the top and down the side, it is time for the eggs and sperm to come together to make the babies. Remember, the boxes are representing all possibilities for the babies. And just like before, all the ones sitting up top have to go down that column, and all the ones sitting on the side have to go across to every spot in that row. And you could do this two different ways. So here, this R would come down, this R would go over, this Y comes down, this Y goes over. So you can write it like that. The R's have to be together and the Y's have to be together because the R's are the gene for shape, the Y's are the gene for color. And the shape does not affect the color. So you do not want to have a RY sitting next to each other. The other way you could write this would be like this. So you bring this R down, this R over, this Y down, this Y over. So you could write it as one big genotype, or you could split them up like I did in the top. You'd fill in all the boxes, and then you'd have to go around and say, you know, is it round yellow, round green, wrinkled green. We're going to practice some of these in class. Here's all the possibilities once you've filled in the boxes. So there's round yellow, round green, wrinkled yellow, wrinkled green. And this is, remember, was originally his F1 cross, heterozygous crossed with heterozygous. And what Mendel noticed is when you do an F1 cross with a dihybrid, you always get these results. 9 out of 16 times you're going to have round yellow, which were the dominant traits. 3 out of 16 times you're going to get the round green, where the round's dominant and the green's recessive. 3 out of 16 times the wrinkled yellow, where the wrinkled's recessive, the yellow's dominant. And 1 out of 16 times where you have the two recessives together. So you always get the ratio of 9, 3, 3, 1 when you're doing a dihybrid cross with the F1 crossed with another F1, or heterozygous crossed with heterozygous. Here's just another example actually showing you physically what they look like. Okay, so we have a P1 cross. Mendel's P1s are always pure crossed with pure. Here's the example of an F1 cross, heterozygous with heterozygous. So if you want to put it into words, a homozygous round plant, heterozygous yellow seed is bred with a heterozygous round, homozygous green seed. That's how you would describe the P1 cross. If you were to describe the F1 cross, you would have a heterozygous round, heterozygous yellow, crossed with a heterozygous round, heterozygous yellow. From all of Mendel's work came Mendel's principles. Here are some of them. Inheritable factors or genes are responsible for your characteristics. Genes are responsible for your traits. Phenotype is based on the genotype. In other words, your genes determine what you look like. Each trait is based on 2 genes, one from the mom, one from the dad. So your trait, you got a gene from mom and a gene from dad. True breeding individuals are homozygous, or both alleles are the same. Okay, that is where we're going to stop today.